joining us today for this special session in advance of launching the first Connectivity Map Challenge. Beginning this past winter and running through spring 2017, Topcoder is hosting a series of data science and algorithm challenges to explore problems in precision medicine. These challenges are brought to our community by the Crowd Innovation Lab at Harvard University in collaboration with the Broad Institute and funding from the Kraft Family Foundation. As you may know, the first challenge in our series was tasked with developing a faster algorithm for DNA sequencing, and it wrapped up last month. This week, we're excited to launch the second challenge in the series. In this contest, our first of a series of connectivity map challenges, competitors will be asked to bring new ideas to improve solutions to the challenge of using 1,000 landmark genes to infer the gene expression profile of a remaining 19,000 in the human genome. And joining us today to answer questions about the background, biology, math, and computer science of the challenge is Aravind Subramanian and Ted Natoli of the Broad Institute. Also joining us from the Topcoder Data Science practice is our own Jen Odess, who will be moderating this session. And I'm Andy Lamora, handle Beachops. So, Aravind and Ted, from all of us at Topcoder and the Topcoder community, welcome. And with that, we'd like to turn it over to you to tell us a little bit about the problem.
Hello. Thanks for the opportunity to present on our work as part of this Topcoder Challenge. This is Aravind, and I have Ted Natoli, a colleague, and we'll tell you a little bit more about connectivity map in the generic and also in particular for the upcoming contest. So, connectivity map, just to start off with a little bit of background on what connectivity map is. So connectivity map is a resource that we're building at the Broad Institute of MIT and Harvard and Cambridge, in which we are cataloging a genome-scale library of cellular signatures representing the responses to chemical, genetic, and disease perturbations. We have collected a large data set, and this data set is being used across numerous biomedical labs across the world. And we'll tell you a little bit more about what we do and what the contest is about. So diving in, the goal of the connectivity map, as I mentioned, is to construct a library of cellular signatures. So by a library, what we mean is a data set that is comprehensive, which is to say it has every possible gene expression profile that one could make, just like the Library of Congress may have every book that's ever been published. We capture the action of small molecule drugs as well as diseases on biology in the form of information-rich readouts. We'll tell you a little bit about that. But importantly, these readouts are not optimized to a particular disease or a particular cell type. Instead, they apply equally, the concepts apply equally to any disease or any cell type that one might want to research. The resource we're building, importantly, is used by biologists across the world, many of whom are not particularly sophisticated with analysis, and hence it is important that the resource that we're building offer means to easily look up results, look up data. Much like you can search Google for information, we imagine that the connectivity map is a resource where you can easily search for perturbations that match your particular disease of interest. It must be easy to integrate multiple data sets, so we of course are not the only people in the world who make such data. There are other folks, of course. We make it at a much larger scale. However, there are a large number of other labs across the world that make smaller data sets, and it's important that it's easy to compare data made in smaller labs with data made at our center. And of course, all of this put together must be accessible to all people in the biomedical community, which includes biologists as well as computationalists such as ourselves. So that's our goal, is to build such a resource. I want to start off with describing what a connectivity map experiment is. So unlike other areas of biology where you have lots and lots of complicated experiments, in connectivity map there fundamentally is one kind of experiment, which we call a perturbagen-based experiment. This starts off in the following way. We assemble a collection of perturbagens. A perturbagen simply is a fancy word for a drug, which is to say a small molecule chemical, or it could also stand for a genetic reagent that we introduce into cells to manipulate the genetics of that cell. So these are perturbagens, categorized broadly. We add these to cellular cell types, which are essentially cells representing various kinds of lineages, but that are able to grow on plastic. So we add these perturbagens to plates that contain these cells. These perturbagens disrupt the biology going on in the cell, and hence they create a response, which we then quantify using an assay in which we measure gene expression. The name of our assay is L1000, and we'll say a little bit more about that as the talk progresses. So, in summary, a CMAP experiment consists of doing this particular schematic that I've depicted many, many times. So we do this hundreds of thousands of times, and we collect a lot of data. Each experiment represents the reaction response of a given cell type to a given drug, and we catalog all of this in a big database. Now, once all this data is collected, well, what do we do with it, or what do folks in the community do with it? Well, there are at least 4 or 5 main applications of it. One important application is to understand how drugs work, so to speak, the mechanism of action of drugs. What is the mechanism, what is the biological pathway by which drug X is effective for disease Y? So this is a standard question that the connectivity map is useful for. Other kinds of questions include structure-activity relationships, which is to say that people who discover drugs or who come up with drugs typically analyze the chemical structure of a compound to assess its impact on biology. Once one collects a large data set of this sort, it is possible to analyze those data to assess what it is about a particular chemical structure that creates a particular kind of biological activity, and this information is useful for the development of future therapeutics. So our data set is amenable to such discovery. Importantly, it's not just for drugs. We also our data set is also useful for people who seek to annotate the genome. So while the human genome has been sequenced for a number of years, the function of the biological function of many genes, perhaps most genes, is poorly understood. In our data set, because we go and synthetically modulate the expression level of every gene in the genome, it is possible to use those data to come up with hypotheses as to what a given gene does. And the technique we use here is a guilt by association, and I'll talk a little bit more about that. Of course, it's not just the gene that matters, it's the variants also that cause disease. For example, people with particular forms of disease have a form of a gene that is different from the wild type. We can apply our methods to understand these variants also, and also the impact of these variants, the differences between the variant and the wild type of given genes. So there are a number of applications, all of which are geared towards understanding how biology works in terms of the genetics of a cell, as well as how those genetics interact with therapeutics developed by the small molecule and drug discovery community. Okay, so these concepts, of course, are things that we work on, but they're also worked on by a large number of other groups. Our fundamental approach to this consists of making large data sets in which we profile the action of these drugs on cells and catalog that using a gene expression assay. The problem with this assay was that it's expensive, which is to say that doing this at scale across thousands of drugs and thousands of cell types is a problem that would take up a lot of money. It would be very expensive to do this using conventional mechanisms, conventional platforms of measuring these responses. So what we've done is we've come up with an assay that we call L1000 that is based on the following premise. Even though there are 20,000 genes in the genome, not all genes do the same thing. Not all genes do different things, which is to say that genes often cooperate with each other in so-called gene clusters, and they travel together to perform a particular function of a pathway and so on. So our notion was, well, rather than measure every gene explicitly, perhaps we can get away with measuring a reduced representation of the transcriptome, the so-called landmark genes, and then use relationships learned from whole transcriptome data, a model essentially, to understand, to extrapolate the 1,000 genes that we actually measure in our assay to all 20,000 genes in the genome. So in other words, we initially collected a large data set of whole transcriptome profiles of all 20,000 genes. We then analyzed that data to find genes that often travel together and hence are highly redundant. We used an approach called using principal component analysis to come up with a set of genes. We did a simulation as shown on this slide, which suggested that if we measured 1,000 genes and then inferred the rest, we could recapture about 80% of the information that was available when we actually measured every last gene. So if one could then measure these 1,000 genes at high throughput, low cost, one would be able to then get 80% of the information that one gets with the whole transcriptome profile, but at a much lower cost. So that's the premise of what we worked on. This technology is called L1000, and we're using this in a number of studies, and this contest is based around the notion of improving this inference. So we're going to come back to the details of the contest shortly, but I just want to talk a little bit more about how the data set, once it has been constructed, is used, and then we'll come back to the specific contest. So I'm going to skip over the details of the assay, but just to point out that the L1000 assay is a molecular biology assay that involves a large number of lab steps, many of which we've automated, but it still works with conventional molecular biology techniques, including the need to amplify mRNA derived from whole cell lysates. We then detect these so-called amplicons on instrumentation from a company called Luminex, which we've adapted in a number of ways. But the net effect of all of this is that the cost for us to make data is dramatically lower than using a commercial platform. We have large amounts of automation in the lab that go into the act of making data at high throughput. We have various kinds of fluid robotic stations, and this really is a resource that that a number of labs across the country and the world depend on for data of this sort. So the impact of these data is much more than just at the Broad. It is likely to be across many other labs. All right. So what does it actually mean? So what does it mean when you and you'll see aspects of this data in the as part of the contest. But I just want to walk through what making a data set means in this space. So as I mentioned, the experiment starts off with a set of perturbagens. A set of perturbagens, which could be small molecule drugs, or could be genetic reagents, all of which we acquire in a 384 well plate. We then treat cells of different lineages with this set of perturbagens, which elicits a response. We capture that response in an L1000 profile, and we save it in a large database. So, for example, just to walk through the process, if one had one perturbagen, and one profile is one perturbagen in one cell type at one dose, and for one duration of treatment, that yields a signature. So one signature is derived from a perturbagen in a cell type and at a dose and a particular duration of treatment. If one does this now for, in this example, 348 compounds, and one does it across 12 cell types, 6 doses, you can see how it mathematically computes to about 25,000 such signatures. So in our system, we routinely make about 4 times this number of signatures a week, and hence this data set tends to grow at a pretty rapid clip. All right, so once we make the data, so of course, in connectivity map, making the data is just step 1 of the process. The part that follows the act of simply making data is to process these raw data files to come up with the numbers that are actually used in analysis. So again, just in brief overview, the process starts with scans. This is what I call raw data, which gets deposited on servers connected to the lab automation equipment. We then apply a number of steps to normalize the data. What normalization means is to ensure that the amount of, because there is some variance in the amount of mRNA that every well in a plate sees, just because of the various automation and various steps in the process, one can never control that exactly. So we have to adjust for that computationally. So what we do is we have a set of control genes, around 80 control genes, that are measured as part of the assay. we use these control genes to make sure that the amount of signal coming from every well on a plate is about the same, and hence this process is called normalization. Importantly, what happens after this is the part that is most relevant to the contest, which is to say that we measure only 1,000 genes, so our initial readout of a normalized experiment is 1,000 genes. We then apply a pre-existing inference model to extrapolate the expression values of the 19,000 odd other genes that are not measured. Actually, the number is not 19,000; it's about 12,000. It's a technical detail which we'll get to later. But the concept remains the same, which is to say that we extrapolate 1,000 genes into the whole genome transcriptome profile, which is then used in all analysis. So post the inference step, which is what this contest is about, we analyze the data by computing differential expression, which is to say genes that are up in a particular treatment compared to a control. We then concatenate multiple signatures to make a consensus signature, which represents a robust signature of a particular perturbation. And then, of course, most importantly, we use these data to compute relationships between perturbations, and that particular activity will be in a future contest, will be a topic of a future contest. But focusing on the use of the data, once we come up with this graph of relationships, we can then query or interrogate these relationships in much the same way as you can query Google with a set of keywords for a question of interest. So in the CMAP world, this is how a query happens. As I mentioned, the matrix, the data set is a massive matrix where every column is an experiment. We currently have close to 2 million such experiments. And every row is a gene, of which there are about 20,000. The first 1,000 of these genes are the ones we measure in the assay, and the remainder are ones that we infer in the manner I just described. So with this data set, well, we ask questions in the data set in the form of queries. So one would go to every column in this particular matrix, and one would ask the question, is a set of genes that one is interested in, are those set of genes triggered in a particular column or not? One would then compute a score for each column, sort the column, and return that list to the user, where the top rank column is the column that most recapitulates the expression, the up or down regulation of the genes that the user put in. So that's called a query. Here's an example of a result that comes from such a query. When a user here had queried our resource with a list of genes that they had done in their lab that were up or down regulated by BEZ235, a known PI3K mTOR inhibitor. When they queried our data set, this screen shows a snapshot of the top results. Most of the top compounds returned in response to this query are members of the mTOR PI3K pathway. The implication of this is if the user had not known that the compound that they were querying with was a PI3K mTOR inhibitor, they would have derived that information by simply looking at this list. In other words, guilt by association. Similarly, you can the another cool thing about this data is it's not simply restricted to compounds, which is to say that you can query with your compound of interest, and you can come up with other compounds that do the same thing. But importantly, in addition, you can find genes, meaning genes in the transcriptome of the cell being profiled, whose loss or gain of function phenocopies that of the compound. So in a disease, if you knew a particular gene was mutated or dysfunctional, you can then use that information to query our data set to come up with a potential therapeutic that would reverse the expression level of the disease gene. So this is kind of why it's connectivity map, we believe, is a particularly exciting resource for therapeutic discovery, personalized medicine, and so on, or precision medicine. The notion there being that one can bring a particular gene expression query. It doesn't matter what platform, doesn't matter what technology was used in it, but just the same way as you would type in a set of keywords that you derive some of at Google, as long as you can have a list of genes that are representative of the cellular state you're interested in, you can query the resource and get back a list of compounds and genes that phenocopy your input query. Here's another example, which again speaks to the fact that the connectivity map concept allows one to walk between compounds, genetic loss of function and gain of function. In this case, when a user queried our data set with a gene expression profile of trametinib, which is a known MEK inhibitor, they got back from the system a set of compounds, most of the top of which were all known MEK inhibitors. Again, if you had not known you were querying the MEK inhibitor, you would have probably figured that out once you saw this result. You can also extend this to genes where either the knockdown of the gene or loss of function correlates with your input query, or also importantly in this particular case, where overexpression of a given gene also phenocopies the act of the compound that was used to treat the cells. So putting all this together, if you want to shut off a pathway, for example, that is triggered by one of these genes, you would then query this database and come up with a list of compounds. And because compounds are simply more therapeutically easier to administer in a clinical setting than a genetic reagent, at least as of now, compounds are more powerful in that manner from a therapeutic discovery perspective. All right, so as I mentioned in Connectivity Map, we make a lot of data, we have algorithms, and we continue to improve the algorithms, including for this contest. But it's really important to remember that the resources we are building here, and hence any improvements that the community makes to these resources, are going to impact not just our lab, but are going to impact the hundreds of labs across the community that use our resource. And because of this, we have an effort in which we make all these complicated algorithms accessible to bench biologists in a web browser. So even though the algorithm under the hood might be pretty complicated, may need lots of compute infrastructure to run, the tools that the user sees is refined and is much more user intuitive, and it's because of all the effort that gets put into making these tools easy to use that we have a large community of people who are able to use our data set who in principle can benefit from any improvements to the algorithm without necessarily having to understand every last bit of mathematics that went into making those improvements. All right, so I'm gonna probably end with a example that is perhaps the most relevant to precision medicine, which is in this particular situation a data set in which we extracted from a NIH data set a set of samples got from individuals that were on a particular phase 1 trial for a CDK inhibitor. In this particular experiment, we had access to data from their baseline biopsies. These patients were then treated with PHA 793887, which is an investigational CDK inhibitor. And then we also got data for these same patients a few months after they had the therapy. In our analysis, and then with these gene expression signatures, we queried the database, the Connectivity database, to see if we could understand why some of these folk were able to respond to the treatment, and why some of these folks were not able to respond. And what we noticed was that the rank of the target of the particular investigational compound that these patients were receiving, the PHA 793887, the target of that, CDK4, was engaged, meaning it was diminished in people who did respond, and this is statistically significant, while it really was not particularly diminished in people who did not respond. The implication of this being that perhaps it was a lack of target engagement between this particular compound and CDK4 that caused the people who didn't respond to not respond, and it's not quite a situation where it was on target but didn't have the effect. In this particular example, yes, it's a small data set, but simply extrapolating on the basis of the small data set for the sake of an example, the insight that Connectivity Map gave investigators here, clinical investigators here, was that perhaps the reason why people didn't respond to this therapy was because their particular genetic mutation simply didn't engage well enough with the drug that they were being given. So in principle, if this particular data set or this particular approach continues to pan out, one could imagine that a profiling effort of this sort could give immediate feedback to people in the clinic, to give immediate feedback to their care providers and the patients to see if a particular therapeutic regimen that they're on is working for them in their particular situation or not. So this is, of course, quite preliminary. This is obviously research, but it has potential to make an impact in this particular way. So I'm just going to end with the fact that we have a large team of around 30 people at the Broad Institute who work on this project. We build a number of resources, all of which are cloud-based, and much of this, parts of this that will be part of this contest. And we have a program where we take the best results got from algorithms of this sort and package them into intuitive web applications that folks around the world can access and use. I'm going to now transition. So this is, I've been talking a little bit about the background of connectivity map, the concept, and how these data are used eventually. We're going to just transition now over to talk a little bit more about details about the particular first contest, which speaks to the very initial part of what we do, which is to say the inferring the expression values of the genes that are not explicitly measured. Obviously, any improvements to this particular algorithm will impact the entirety of the data set, and hence we think it's an important challenge. So I'm going to hand over control to my colleague Ted Natoli, who will walk you through details of the contest.
Hey, Aravind, can you guys hear me okay?
Yes.
I wanted to say, too, before you pull out of that, that presentation, there was actually, I believe it was slide 14 or 15. It was where you were showing the ranks. And there was a quick question. It looks like it might be 13 even, possibly, if you'll just go back. That one, yep. So the question is, how do you rank these genes, the p-value or fold change?
So these scores here represent the extent of connectivity, which is a measure of correlation, between the particular compound shown here and the input query. So as I mentioned, each one of these drugs is one column in this large, massive matrix. Hence, the input query, we compute a correlation score between the input query and each one of these columns. We then summarize that across cell types and across perturbagens to come up with a so-called summary score that is depicted out here. So in this particular table, while we show the summary, it is possible to drill in and find the specific connectivity score in a given cell line. So we typically do an experiment in 10 cell lines. The summary is the summary score of all across all 10 cell lines, but it is possible to drill into a particular cell line. So I should point out that this act of finding similarity between a query and a profile in our database is a very important problem. It's not quite the topic of this first contest, but it is an important problem, and we currently use a correlation-based metric to compute it, and that correlation-based metric depends, in many cases, on inference of the 19,000 odd genes we don't measure to be as good as possible for that to work well.
Okay, thank you. Yeah, and I didn't mean to interrupt. Feel free to pass it back over to Ted, but just wanted to get that question answered.
Perfect. All right, so I'm just going to, so Ted Natoli is going to walk us through the rest of this.
Thanks. Okay, so I'm going to talk a little bit about the specifics of the contest, and just to sort of reiterate on the importance of this again, as Aravind mentioned, a lot of the value of our data set comes from being able to compare external users' gene sets of interest with L1000 data. So L1000 is a great platform, but there are many other gene expression platforms out there, so a lot of the world's gene expression data is not made on L1000. So in order to be able to compare L1000 with these other data sets, having high fidelity inference values is very important. So just to talk a bit about the specifics of what we're aiming to do here. So in this contest, our thought is to provide the contestants with a large training data set of publicly available gene expression profiles generated on a particular platform that here, just for simplicity, we're going to call Platform A. So that's this large gray box here in the left. In addition, we're going to provide about 1,500 samples of L1000 data. And these samples are interesting because we were able to profile them both on L1000 and on another contemporary gene expression profiling platform that measures all genes in the genome. And we're going to use that platform here, for convenience, just labeled as Platform B, as our ground truth. And so the idea would be that using these 100,000 samples and the identities of the 970 landmark genes that L1000 measures, contestants will be able to devise some kind of an algorithm to predict the values of all the about 11,000 genes that are not directly measured by L1000. So with these three data sets, our thought is that contestants will be able to iterate offline, building and training their models, and then using these 1,500 or so L1000 samples with the ground truth sort of answers to be able to refine and improve their algorithms. And once an algorithm is ready for submission, in this particular contest, the format is submission-based, so all they'll need to supply is essentially a matrix of the inferred gene expression values by all the samples. And what we'll do over here in this provisional test scoring phase is take the user's inferred expression values and compare them to the ground truth values, which we've generated by measuring them directly on another platform. And based on how well the inferred values replicate the ground truth measurements, that submission will receive a particular score. And so that can happen iteratively throughout the duration of the contest. And then finally, at the end, we'll perform the same sort of measurement, but the same sort of assessment, but on a separate set of holdout samples that have not been provided to the contestants until the very end. And to talk a bit about how the scoring is done, one approach we've taken is that for a particular gene, if we want to assess how well we're inferring that gene, we can take its expression profile across an array of different samples. In this case, we've used about 1,000 of these samples where we've got both L1000 and non-L1000 ground truth data. And for every inferred gene, we can compute its correlation with itself across those two data sets. So you take the inferred gene across all of its samples in the L1000 data set and compare it to its measured equivalent in the ground truth data set, and you'll get some correlation value. So you can do that for all 11,000 or so inferred genes. And also with that correlation, you can convert it to a rank by asking how well does the inferred gene pull up its measured equivalent. So it could be the case that the absolute correlation isn't that high, but the particular gene's pattern of expression is unique enough that it's able to find itself well across those two data sets. So the rank could be still good, even if the absolute correlation is not that high. So this plot here on the left is showing both of those two measures. So the x-axis is the correlation, the absolute correlation between the L1000 inference values and the measured ground truth values for each gene. And you can see that the distribution is sort of wide. It's centered around 0.35 or so, which in an absolute sense is not extremely high. But if you look at the other dimension, which is the rank, you can see that the ranks of the genes are typically pretty good. There's an array of correlations that correspond to a high rank, but the ranks in themselves are actually pretty good. I should point out that there is a large number of genes, you can see this large tail here, where the ranks are not very good. So those are the genes where our current model is not performing very well and where there's room for improvement. And we're hoping that this contest will yield some improvements for those genes in particular. And to summarize these two measures for each gene, we can compute an average, essentially, between the correlation and the fraction rank. And we feel that these two things are fairly equivalent measures, and so simply averaging them gives us a score for each gene. So theoretically, the best you could do for a particular gene is a score of 1, and our current algorithm gives a median of about 0.68. So it's okay, we think, but we think there's room for improvement, and we're hoping that this contest will give rise to some improvements, especially for the genes where we're not doing such a great job. Are there any questions on that?
I think that's pretty good so far, Ted. I know we have a lot of questions in our Q&A document to still get to, but I think that was very helpful. Do you and Aravind have any more slides to speak to in the presentation?
No, that's it. That's it. We're all set.
Awesome. Well, I just want to say a really big thank you to both Aravind and Ted from the Broad Institute for taking the time out, first of all, to give this kind of expertise and level of detail to our community. I think it's really valuable, and if this format, this kind of webinar discussion is well received, I'd love to be doing more of these. And so the session is by no means done, by the way. We still have about 30 more minutes left to do Q&A, and we have a lot of great questions to ask, but I just want to say a special thanks, both Aravind and Ted, for taking the time. And also introduce myself, for those that don't know, I'm Jen Odess. I run our data science practice, so many folks probably have seen me in your newsletters, in the forums, emailing many of you. So thanks for everyone who is participating, and like I said, I hope you find this format very useful. The CMAP Marathon Match is coming up really soon, so in this particular discussion really lends to that match. So you should find it quite useful, and if it's not too late to get your questions answered, you can put them right into this Hangout. And we'll make sure we'll do our best to get them answered for you. And then also for anyone who is looking to re-watch this session, it's going to live in the Topcoder YouTube channel, as well as we're going to send out a really nice blog summarizing all that we learned today. So there's plenty more options for everyone to be able to see all that we've taken away from today's discussion. So with that, let's actually hand it over to Q&A. And Andy, I think you've got the first, actually, you know what, I apologize. I want one quick question. So Sun in our chat window had asked on that very first question, Aravind, when I had you go back to the gene rank slide, he asked how the input gene list was actually ranked. And I'm not sure if we specifically asked it that way or not.
Yeah, that's a good question. So our current algorithm actually does not rank the input gene list. It's simply treated as a bag of genes. So it's a bag of genes. So there's no ranking inherent in the input query. Obviously, our database does contain genes that are ranked, where the gene that is ranked the highest, ranked 1, is the gene that is most different between treatment and control, and the gene at the end is the other way around. But the input query, we consider it to be a bag of genes. And it does not come with any score or a rank.
Great. And thank you for answering that. Aravind, can you also see, if you click the Q&A tab to the left of your screen, do you see there's a handful of questions? There's one of our members just posted a question. I think it'd be easier for you to read it. Oh, you know what? Sorry, Clinton. Can you take off Aravind from the presenter? Awesome. Thank you. So hopefully Aravind can see the question from, looks like member Tao. You see it?
Yeah, we're just trying to read.
Oh, no problem.
Yeah, so I'm not completely clear, but it seems to me that the question being posed is one on what is used to infer the target gene. Right. So this is a good question, and I think the formula just might be getting a little mangled, but I think the intuition behind this is right, which is to say that the model takes the 978 landmark genes that are measured, multiply a linear regression model will take these 978 genes, multiply each of these by a weight that is derived from a training data set, so as to infer the expression level of a gene that is not the 978. We happen to use a particular way of doing this, but what this contest is about is obviously coming up with a better way of using these 978 actual measurements to predict the value of a gene that is not measured. So brief answer is yes, W0, W1, those are the weights that we use in our model. But I can't talk a lot more about that because this actually is what the contest is about.
Great. Okay, thanks. And Aravind, if it's easier to go back to your slides so that you can have them to reference as we go through the next handful of questions, feel free to share back that screen.
Sure.
Okay, so I think we're gonna. So by the way, everyone, keep putting your questions into the chat window, and I will be the one going and pulling them and kind of interjecting to make sure we get those in. In the meantime, I know we have a couple really important questions that Andy wanted to ask, so I'd like to hand it over to Andy to start with those.
Thanks, Jen. All right, we have a list of questions that came in from community members and others prior to the start of the webinar. We collected them over the week. So, Clinton, if you're able to share it on the screen, it could be helpful. Otherwise, I'm just going to jump right in.
I could probably do it too.
Thanks, Jen. I think that'd be a good idea.
No problem.
So the first question is an easy one. What is the current state-of-the-art method for solving this problem? What techniques are used? So if you were teaching a master class, what would you point people towards first?
Right. So what we have is, and what we're providing as part of this is a training data set that contains gene expression measurements of all 20,000 odd genes. I forget, what is the exact number of genes in the...
It's about 12,000 unique genes.
It's about 12,000 unique genes, not 20,000. This is just a technical detail. So whenever I say 20,000, think of it as 12,000 because that's the size of the transcriptome for this particular configuration of the data. So what the current approach is to use a training data set of these 12,000 genes measured across 100,000 samples. We know the identity of the 978 genes that are being actually going to measure on on new data. Hence, what we currently do is to infer relationships between each of the 12,000 minus 978 genes and the 978 genes. We happen to use all 978 genes to do this particular inference prediction. But of course, there could be alternates to this. But to address the question, we use each of the 978 measurements. We build a model on the training data, and we use it to infer the value of each of the 12,000 minus 978 genes.
Thank you. If I could drill into that a little bit further, and it may come up in some later questions here, though, but are there specific computational techniques that are understood as sort of the opening play on the chessboard, or common approaches to working on the problem? So when you're processing it, not just the biology.
Yeah, sure. I'll mention two, and both are in there are papers about related papers in both of these, so this is public information, so to speak. One method we use is simple linear regression. So we simply build a model on this training data set between a gene to be inferred and the 978, and hence we get 978 weights representing the linear relationship between each of the landmarks and the to be inferred gene. That's one approach. There are also some approaches that use more sophisticated deep learning approaches. This is highly preliminary, so I think if I were to so those are areas we've just about begun to explore. I'm not completely confident as to how effective they will be in this particular scenario, but that's kind of what this contest is about.
Got it. Thank you. And any follow-up material we receive, we'll be happy to post to the forums and challenge and refer to them in our blog for those of you following from home. Next question I have is coming from a member. In fact, I'm going to combine two questions here a little bit from B. Turing and KS Pham. The question is, in L1000, you aim to get 1,000 genes that their expression can be used to infer the remaining expression of the other genes. Does this set of 1,000 genes change depending on the cell type?
Yeah, so this is a very good question. Yes, the short answer is yes. That's we do the measurements of 1,000 genes in every cell type. We do the profiling experiments in multiple cell types. So yes, some perturbagens produce about the same response in every cell type, but a fair number of perturbagens do give different responses. The consequence of this is, of course, that the 1,000 measurements of 1,000 genes does vary between cell types, and that's a factor to be taken into consideration in this contest, because both the training data as well as the test data is not from one cell type, but they come from a variety of cell types.
Yeah, and just to add to that, the approach that we've taken is to devise one model that we will use in all cases. So it's fairly generic, but it's a good point that it's possible that the 978 landmark genes may not behave the same in all cell types. So one potential way that our algorithm could be improved, perhaps, is to sort of weight them differently depending on what the cell type is that they're in. And so there may be some empirical way in the training data to detect potentially clusters within the data set that sort of go together based on their baseline profiles. And there may be some sort of cluster-specific effect that you could incorporate into the model, as opposed to just having a single set of weights that are applied in all cases.
Thank you, Ted. So that might be an answer to the next question then from KSFAM, which was, how are you covering all these different types of CMAP? And if I understand you right, the answer might be it's up to the choice of the competitor.
Right. Yes. And it also is the case that the cell types used in the training data are sort of reflective of the cell types that we're going to use in the evaluation data, assessment data that is held out, and hence models built in the training data, should easily apply to the holdout data. But as Ted was pointing out, it could be that an ensemble of models built per cell type outperforms that of a single model. This is perhaps something to be looked at.
Thank you. So the other half of that question is more of a biology and practice type question. So if you are working on different cell types, and there are so many different cell types, how are you creating drugs to target them? Like, what's the pathway from just selecting which type to work with and where it could end up with treatment?
Yeah, yeah. So that's a good question too, and just maybe a two-part answer to this. First is there are obviously a large number of biological processes that are shared among multiple cell types, right? So there is a large commonality, and of course for those ones, it doesn't matter. It perhaps doesn't quite matter so much which particular cell type you do your analysis in, because it's about the same. It's biology that is shared. In such a situation, an experimentalist would pick a cell type that is most easy to work with in the lab. Of course, there are aspects of biology that are very cell type specific. Perhaps there is a receptor that is only expressed in some cell types and not the others, and the entire response of a cell to a given treatment is dependent on expression of that receptor. So that's an important concern. There certainly are a number of drugs that act in that particular way. To address this, our data set spans profiles across multiple cell types. So we already have close to 15 different cell types in the data set, and that this particular angle is being continually improved. And over time, you can imagine that we're doing connectivity map across a whole range of cell types, thereby covering, as much of biology as we can. I think it's pretty good already. And cancer cell lines, which is typically what we use, are a powerful tool for a range of diseases, both cancer and non-cancer. So I think the short answer is our current data has about 15 cell types. The longer answer is that, well, that's a good start. And in time, we could imagine expanding this to even more cell types to make the data set richer.
That's great. Hey, Andy, we have about, I would say, maybe five to 7 more minutes of questions we can get in. Then I want to do a few closing statements before we wrap up at the top of the hour. You may be muted.
I might be indeed. Thank you, Jen. So thank you, Aravind. And back to some questions more about the teamwork environment at the Broad. And we haven't really explored that in this call, just how special a place it is. So feel free to expand on this a little bit, too, to talk about the different parties involved at Broad. But are there other academic groups in the Broad that you were collaborating with, and can you give an example?
Yeah, sure. I mean, of course, the Broad is a wonderful place for people people like Ted and myself who are trained in computational analysis, because it's the Broad makes these incredible large data sets, and in many ways is a data. It has produced a lot of the data sets that are used in the community. So it's an amazing place to be at, to not just be able to access the data, but to actually be close to how the data set is made. This actually happens to be one of my favorite things about Connectivity Map, is that we do all aspects of it. We make the data, we analyze the data, we make the data usable by others, including via contests like this. So it's a very collaborative environment. I think the second part of your question was on how other groups use data we make. So I think that's an important question too, because Connectivity Map is not really tuned to any particular disease area. The data sets that we make are equally applicable to any disease that you can imagine. Of course, we currently measure transcriptional readouts, so it would be important to establish that a disease one is seeking to address has some impact on the transcriptome, which is the case typically. But because it's at the intersection of so many areas of biology, there are a fairly large number of collaborations that use these data, and a whole varied set of diseases, everything from cancer to rare diseases to neurological diseases that make use of these data sets.
Thanks again. So we have about 8 minutes left, and I know Jen has a couple follow-up questions. But let me ask 2 more, and then I'll turn the mic back over. The next question I have for you is, getting back to the working environment, can you discuss 2 or 3 of the most common and difficult problems that you and researchers are encountering when you work on this type of problem?
Well, I think the certainly one problem is simply the size of the data sets. You can't analyze these data in Excel. You do need to have a compute environment. You do need to have the ability to read in large matrices, to process large matrices. So all our data, including this contest, is basically large matrices. Hence, you need tools that are capable of loading in these large matrices, manipulating these, slicing, dicing, finding patterns in that, and that is typically something that is a little different from the linear flow that one might have one might learn in a computer science class to solve a linear programming problem, linear algebra programming problem. So it is slightly different. I'd say another aspect that is typically challenging, but I think is very important that we address, is to make all these results, even though the underlying algorithm might be complicated, it needs to be, the results of such an algorithm need to be conveyed to biologists or people who are not particularly computationally savvy, or at least don't write code. They need to still be able to access these data, access these tools, and run it not just on our data, but on data that they bring into the system. Hence, to me, I think a second large problem that I think we spend a lot of time working with, and that's important, is to make all these data accessible to both computationalists and also people who work in biology labs.
Got it. Well, then I'm going to add one more question to the 2 that I'm asking here, and then, Jen, I'll turn it over to you with a couple minutes remaining. The first question is, are there other groups trying similar or alternative approaches to CMAP?
Well, so I should point out that the origins of Connectivity Map are in the functional genomics community. Several folks have done a variety of experiments of this sort in other organisms like yeast and so on. In fact, one of the first papers in this field was by a group that did a very similar kind of experiment in yeast. Of course, functional genomics, of course, is also hardly something we came up with. It's what people study often, and they study it by approaches that are very similar to what we use, which means they perturb a cell by knocking out a gene or overexpressing a gene and so on. The key thing that we do slightly differently than what other groups in the world do is we make data at a massive scale. We have this assay that we invented that allows us to radically scale up this entirety of this effort. So the sheer amount of data that we have accessible and that we can make is, I think, what distinguishes the effort here from efforts in other places. The concepts, of course, are shared. I don't think we claim any particular ownership of the concept, but I do think the innovation we've done here is to enable making data at large scale much more tractable, and that actually is exactly what this contest is about. Because if the community can improve the inference of one gene, that particular gene might be a gene very important in a disease, and hence it's very relevant to therapeutically addressing that particular disease.
Yeah, and just to add on to that a little bit, there are some other groups at the Broad who have kind of taken a similar approach to this reduced representation paradigm where even for different readouts, not just gene expression, you can measure a subset of the features potentially and use some model to infer the remainder. So I think if we're able to gain some insights from this contest, those could be applicable to those other assays as well. So it's not just L1000 potentially that could benefit in the long run.
Awesome. Well, with our 3 minutes remaining, I know Jen needs one, so I'll ask for a short answer on this one, but it's one I always like to ask technical folks such as yourself. So, Aravind, Ted, asking each of you, what is your favorite development language and favorite open source library for working on these problems?
Well, I haven't written code in the past 36 months, but when I write code, it was in Java.
And Ted, how about you?
I'm an R user. It's my guilty pleasure. Yeah, I love R. I love all the packages. I'm a big Hadley Wickham fan, as most people probably are. So.
Excellent. Thanks a lot, guys. And Jen, I'll turn it over to you.
That was a great last question, Andy. I'm glad you put it in. That's always a fun one to end on. So all I wanted to end with, guys, just one more time, a big thank you to Aravind and Ted from the Broad Institute. Remind everyone that really the purpose of this meeting, this discussion, was to help benefit any competitors looking to compete in the upcoming CMAP Marathon Match. I have our pre-registration landing page up right here. It's available to everyone. It's crowdsourcing.topcoder.com/CMAP. That's crowdsourcing.topcoder.com/CMAP. Once you're there, it gives you a really nice overview. Oh, hold on, let me get to it. Gives you a really nice overview of the challenge, who all is involved with it, how it's part of the Precision Medicine Challenge series, and then a really nice little a place to input your name, your handle, your email, and to actually pre-register. And this is something new that we're offering. It pretty much gives you a chance to pre-register as early as we make these pages live for an upcoming match you want to participate in. By doing so, you get to stay up to date with direct emails and reminders of when the match is going to launch. And then when the match does go live, you're automatically pre-registered by us for the match. So I encourage everyone to come in here, pre-register for the CMAP contest launching very soon. And again, big thanks to Aravind and Ted and the Topcoder team for making this webinar so successful. And for anyone who's listening either now or who's going to watch it again or later on our YouTube channel or through the blog that we'll be releasing soon, feel free to circle back with any more questions that you have. Let us know what you think of it in the forums. If this is well received, we would love to start doing more of these. So thanks again to everyone, and see you all on the forums. Bye.
Okay, thanks a lot.
Great, thank you. Bye.